Hello everyone, my name is Tianci Song. Today I'm glad to present our work in the ISMB 2024 conference. So the title of our recent work is GNTD, reconstructing the spatial transcriptomes with graph-guided neural tensor decomposition informed by the spatial and functional relations. As we know, in situ capturing method as one of the spatial transcriptomic technologies, it overlays a grid of spot with unique positional barcode over the tissue, and probes of each capturing spot can capture the mRNA transcript from the cells overlapped with the spot, and the gene expression of the cells within each spot can be measured by the existing sequencing technologies. However, there are several challenges preventing the ISC method from being used in the practical studies. The main challenge is incomplete expression profile. The spatial transcriptomic data generated by the ISC method are highly sparse, where most zeros are missing values rather than the true biological signals. Using the incomplete expression might lead to the incorrect or the misleading discovery in the analysis. To address these previously mentioned issues, we proposed a tensor completion method to reconstruct the spatial transcriptomics data from the incomplete expression profiles. With this reconstructed data, we can gain a valuable insight into the tissue biology to further facilitate the biomedical research. Before we talk about our tensor model, I want to briefly introduce the tensor representation of the spatial transcriptomic data. A tensor is generally defined as a multidimensional array with its dimensionality described as order and its specific dimension termed as mode. Due to the specific arrangement for the ISC method, their spatial transcriptomic data can be naturally organized into a three-order tensor with dimension corresponding to the gene and different spatial axes. By modeling raw spatial transcriptomic data as a tensor, the reconstruction from the incomplete expression can be easily formulated as a tensor completion problem, aiming to impute the missing entries of a partially observed tensor. CP decomposition, as a classic tensor decomposition, it learns a linear combination of a compressed representation to reconstruct the observed tensor. However, the interactions in the real-world tensor are often nonlinear, and the multilinear system might not be expressive enough to model these complex interactions. To solve this issue, we propose the GNTD. With the spatial expression tensor in the spatial and the gene modes, the first layer learns the weights representing the linear factors for each mode. And the second layer introduces nonlinear mapping among the linear factors within each mode. And the last layer, the nonlinear factors are aggregated by the learnable coefficient and structured as a weighted CP decomposition, to reconstruct the tensor. In addition, this decomposition is also regularized by the graph Laplacian of the Cartesian product of the spatial neighboring graph and the protein-protein interaction network to further guide the imputation. To validate the performance of the proposed method GNTD, we conducted the experiment on both simulation and real spatial transcriptomic data from TextVidem and StereoSeq. We compared the proposal method with the SOTA method for the spatial expression reconstruction, including the tensor-based model and autoencoder-based method. In the first experiment, we imputed the simulated data with different sparsity by both GNTD and the baseline method, then applied the clustering on the raw and imputed the spatial expression to detect spatial domains. It is clear to see that GNTD consistently outperforms the other baseline method on the simulated data with both low and high sparsity. And when the simulated data is highly corrupted, GNTD still accurately identifies all tissue regions. In the second experiment, we further examined the raw and imputed the spatial expression by GNTD and the baseline method on the highly corrupted data by visualizing the few simulated spatial variable gene. So we can see that the gene patterns recovered by the GNTD match well with the ground truth pattern compared with other methods. To evaluate the imputation performance of GNTD on real spatial transcriptomic data, we first performed a tenfold spot-wide cross-validation for GNTD, and all the comparing method on the 10 different text rhythm data. We can clearly see that GNTD consistently achieved the best performance in the spot-wide cross-validation. Similarly, we also performed the tenfold gene-wide cross-validation for all the methods on the same dataset, and we have a similar conclusion to the spot-wide cross-validation. To check the performance of GNTD in spatial domain detection on real spatial transcriptomics, we imputed both human brain and breast cancer data by using both GNTD and baseline method, then applied the clustering on the both raw and imputed spatial transcriptomic data. We observed that GNTD generally showed superior performance in spatial domain detection compared with other baseline methods on 2 annotated tissue regions. In the last experiment on the real spatial transcriptomic data, we imputed human brain data using both GNTD and tensor-based method, and then visualized raw and imputed spatial patterns of cell marker genes, and compared them with the corresponding in situ hybridization images. It is obvious that GNTD can properly impute the gene expression with continuity and enhance the spatial pattern, highlighting the corresponding tissue structures. We also applied the same experiment to the high-resolution spatial transcriptomic data seq. Please see more details in our poster or the published paper on Nature Communications. And thank you for your listening, and please let us know if you have any questions. Thank you.